So I had a colleague in New Zealand who gave me this analogy: what's the difference between research and innovation? And so I talk about kind of the research and innovation continuum for a moment. So his definition of research was, research is when you take money and you turn it into knowledge. And the cap there is my representation of knowledge, and it could be, publishing scientific papers, the posters, conference like this, great graduate students, PhD students graduating, postdocs, personnel to go and work at places like CDN, for example. That's all part of the research machine, and that's really important. But then there's another phase, and that's how when we take that knowledge and we turn it back into cash. And that is basically innovation. So the whole continuum is really important. Obviously, we want to invest in research because it's going to have an outcome for the industry. And I think as researchers, it's that first phase is the easiest part. That's like a 4-lane highway. Graduate the students, you publish the papers, you write the grant reports. The next stage of the innovation pipeline, I'd say, is a bit more like just a 2-lane road. It's a harder road, I think, to take all that information now and actually turn it into something producers use to make change. So I spent a lot of my life so far at the top end, trying hard at the bottom end, but now the focus really is at that bottom end. So it's easy to get confused. We have the American Angus Association. I work for Angus Genetics Inc., which is wholly owned subsidiary of Angus. It's a for-profit company. And the idea there is we don't just work for American Angus, we can work for other breeds as well. Strategic partnerships. I think one thing I want to do, and that's looking around this room, great scientists, we need to work with those people. We need them basically working on projects that we can use in Angus. So we do genetic evaluation services for other breeds, Canadian Angus, obviously, joint evaluations with us, Canadian Charolais, American Charolais, Senepol, and a small plug, always open for business. So the selection tools that are available to breeders, there's a long list, and traits get added all the time, and I'll go through them in a bit more detail, but we've got production traits, maternal traits, carcass traits, and then dollar values, which are indexes, putting all the traits together. So you might think, well, Angus is the biggest breed, and we saw 1 billion pounds of certified Angus beef sold last year. So I think back to those early Gentech meetings that we had, and we talked a lot about, can we get genomic selection work in beef cattle? How big does the training population have to be? Is it 1,000? Is it 2,000? How many animals do we have to have genotyped? That's a boat leaving the dock, okay? So that's heading away from us. And my point here is really that the ship has sailed on genomics. Like, it is up and running, and it is working in beef cattle. So I don't think we have to answer the question anymore about does it work. So how important is it that breeders keep taking measurements? And that's a question that comes up all the time. And the point is we can't train genomic predictions without data. That's just a fact. It's all about the database, and this is really the key here is the database. Millions of records. It's fair to say it is a transformative technology, unprecedented change. Back in 2010, we're still talking about, is this actually going to work? And now I don't think there's a question that it's going to work. Where were we in 2010 when the first Gentech meeting was around? Just 2,000 animals genotyped. That was kind of the first calibration, and that was kind of a big deal in 2010. But it wasn't a big deal for very long. And I remember at the 2011 conference, I had a slide that had a chicken and an egg on it. And the question was, how many genotypes do you need? And the point was, well, it's a chicken and egg. So once you get a test that works, breeders will use it. Once breeders use it, then you've got lots of genotyping being done. Those animals eventually have measurements, and then you've got a big training population. And that's essentially what's happened. So this latest calibration in April had over 108,000 animals in that training populations. But as I said, if we're going to stay the same, we'll eventually be out of business. So how are we going to get better? And we're going to get better with better data. And one of the areas we want to work on is carcass data. So Mike this morning mentioned ultrasound works, and it does work. Correlation between most ultrasound traits and the equivalent carcass trait is 0.8, around 0.8, which is very high, and that allows us to make a lot of progress. However, real carcass data on the carcass is even better for training our genomic evaluation. So what we're calling the structured sire evaluation is kind of like an old-fashioned progeny test, but now the sires that we're identifying are those high-use sires. They've got a lot of ultrasound data, but they don't have a lot of carcass data. And so they've got a lot of progeny out there. They're going to have a big impact in the population. We'd like to have a better carcass evaluation on those bulls. So right now we have about 2,000 cows bred and targeting another 2,000 this breeding season. And those are coming out of basically cooperating large commercial cow-calf. So the commercial operations, 250 to 600 cows with an AI program already. And it's, sometimes we think about seedstock breeders collecting a lot of great data, but some of these commercial guys collect data every bit as good and professionally as some seedstock breeders. So, but in a way, we're just feeding the same machine. We're feeding the genetic evaluation that exists now. We've already got carcass records. We're just going to add more carcass records. But what about a new way to maybe get some commercial data? And this is kind of an area that I was interested in before we came to Angus. What are some traits we're interested in cows and other places? Things like longevity, fertility, docility we might have on the cow side in the feedlot, and packer side, mortality, feedlot mortality, carcass, health. If you have a steer that's just about ready for market and he falls over dead, that's really costly, and that's a big economic impact. So what can we do about that? So phenotypes are really important. I have to get more creative about getting my phenotypes. I put in beef packing plants. There's a lot of beef packing plants, but there's also a lot of very big beef packing plants, and I think they're a great source of data. And BICS and everything has a lot of potential, and I think getting those data back is very, very important. Obviously carcass data, but I think packers are also a source of other types of data. Data like health data. We had a project in New Zealand looking at pneumonia in lambs. We were scoring lungs in packing plants and have 18% heritability on that trait. So you could score lungs, you could score livers for liver abscesses. You can obviously get carcass data. Another twist that I think we could employ, and this is some work coming out of Australia, has done a bunch of work on this, is DNA, what we call DNA pooling. And when breeders genotype animals now, it's one animal, one genotype. So I got a 50K test, and I genotype that animal for 50K. And at any SNP, he's an AA, AB, or BB. That's his genotype. This idea is actually we're going to genotype now a pool of animals. We're going to take the DNA from a whole group of animals, we're going to put it all together, and we're going to run it over a 50K chip. And now we're going to get a genotype that looks different. It looks like what's the percentage of A and percentage of B in that pool. So just to give an example, here's a couple animals. Here's a group of, I think there's 20 animals there. And there's 2 animals are homozygous AA. These ones in the middle, a dozen of them are heterozygous AB. And then we've got 6 animals over here that are homozygous BB. And when you do the allele frequencies, you'll find that those animals are 60% B and 40% A. So if we did a different pool of animals with a different distribution of genotypes, we'd get a different frequency. So it's a different way of looking at the data. When we first started running the 50K chip, probably back in 2007, I thought, oh, this is fantastic. I can just go into a packing plant now. I can collect all kinds of data, get a sample, I'll genotype the animal, we'll come up with predictions. And one of my colleagues says, yeah, Steve, that's a really good idea. The problem is the genotypes are still just too expensive. So it's just too expensive to collect a carcass record on an animal, a commercial animal, to develop a prediction. But now I'm going to put a whole group of animals together that have got a similar grade, and I'm going to genotype that pool. So again, it's just one genotype, but in this case, I would have genotyped, there's going to be 20 animals contributing to that pool, so the data is more accurate. Is this a delicious steak or is it colossal waste? There's a huge amount of waste on this ribeye, and I use it kind of as a shock factor, I guess. We did quite a few projects in Guelph, and we had great collaboration with Cargill there, able to go in and get rib samples, and we'd sit on the line, and the carcasses would come down, and as you're waiting for your cow to see lots of carcasses roll by, And I was always just amazed at the variation. Here's a carcass, you know, it's stamped, it's going to go to Japan, it's high AAA, it's got hardly any back fat, a beautiful big ribeye. And then you get these other carcasses that come through and they look like this. But there's even some that look like this and they have no marbling. Like no marbling, a ton of back fat. And I'm like, I realize, you know, it's phenotype, is genetics plus environment, but you couldn't take that animal that's got no marbling and that much back fat and feed it any differently and ever get it to have a nice big ribeye and lots of marbling. They're just totally different genetic animals. So that's what we could do with carcass records. But what can we do with a bunch of other traits? And we had a great presentation this morning about foot rot. You know, foot rot. We could be collecting, you know, here's a group of animals. These ones got foot rot in feedlot. These ones didn't. Commercial data on commercial ranches. Guys preg test their cows. Here's a whole group of 2-year-olds that these ones came up empty. These ones are pregnant. You know, that's the type of easy data I think that we can use in beef cattle. Mortality, which ones died, which ones didn't. There's lots of I think applications of this pooled data. This looks like an old picture, another one I got out of the Angus archives. But if you look on the wall, there's a telephone there that doesn't look very old. So my point was that actually auction marts haven't changed very much, and I don't think they are going to change a lot. I think we're going to be, this is how cattle get traded, and they have been since I was a kid, and that's probably not going to change too much. So can we take this DNA pooling idea and turn it upside down? So instead of now using it to get data to feed into the genetic evaluation, I could take, the idea is, could we take a DNA ear punch out of each of those calves? That thing gets pooled together, one genotype on the whole pool, and I get an allele frequency now for that group of calves. That goes through the genomic prediction at Angus, and we come up with, now I have basically the suite of traits predicted on that group of calves. And that's a relatively cheap cost. That's a relatively cheap cost because it's just one genotype for the whole group of calves. And John mentioned this morning about creating the poll for genetics. I think when you have groups of calves with better genetic profiles better genomic profiles, getting a higher return in the marketplace, that's going to drive people to use better bulls, basically. Because I bet you, well, who knows, but this group of calves right here probably has 0 genetic information presented on it if it's a typical sale. So what are some key takeaways? On the application side, it's all about taking the knowledge that we generate through our research projects and turning it back into cash for breeders. We want to create this hockey stick. We're going to do it through partnerships. We've got 4 PhD scientists at Angus where I work. You know, we need to work with PhD scientists here, other universities in the US and around the world to tackle a lot of these projects. Leaders can be replaced, including Angus. You know, if we don't innovate, I think it's we're destined for a not very good place. I think a focus on the carcass has to be there. Again, 1 billion pounds of certified Angus beef. You're not going to ignore that market. That's got to be a focus, and I think the commercial carcass data can help us in that regard. I've got some hope with this pooling idea, and I think turning it upside down and actually putting genetic values on groups of calves kind of closes the loop and could really drive demand for better genetics.